Okay. Thank you. Yeah, originally I was going to talk also about structurally resolved networks for looking at mutations, but actually I think this will be nice and complementary to some of the stuff we've been talking about. Instead, I'm going to focus on anti-tumor immunity and the interaction between the immune system and the developing tumor. So in the paradigm of precision medicine, we're interested in interpreting inherited variation in the context of individual risk of developing disease so that we can better stratify patients according to those that would benefit from early screening or preventative measures. And then what we're talking about here today is more on the other side of precision medicine, which is understanding or interpreting the somatic variation present in the tumor genome and being able, through that understanding, to better map patients to appropriate therapies. And so a lot of these cell mapping efforts are really trying to drill down and understand what's going on here. Something that we've become interested in recently is how does genetic background actually shape the evolution of tumors and the types of mutations that show up during tumorigenesis? And in collaboration with Trey a few years ago, we did a systematic screen using the TCGA for interactions between the inherited genome and the somatic mutation rate of key cancer genes. And we did find a number of interactions, and interestingly, those interactions actually were often trans, and that the locus at which the germline event was often distant from the somatically altered cancer gene. And lo and behold, several of the interactions seem to be converging at the level of biological pathways. And so genetic background affecting a particular cancer pathway may also affect the occurrence of driver mutations within that pathway. So I still am interested in pursuing this direction, but we've gone on a bit of a tangent now, focusing on the immune system. And there's been a great deal of excitement about immunity. We know that the ability to evade the immune system is one of the hallmark behaviors of tumor cells. So clearly the immune system must be putting some kind of selective pressure on tumors. And we know that if we can inhibit if we can interfere with the mechanisms by which the tumors evade the immune system, then we can actually get some quite effective cell killing by the immune system. So this works because of the antigen presentation pathway. Proteins within the cell, mutant or wild type, are degraded by the proteasome. The peptides go to the ER where they bind to this major histocompatibility complex, and they're transported to the cell surface where they can be probed by T cells. And tumor mutations are particularly exciting in this context because they are not expressed in normal tissues and not subject to central thymic tolerance, and so they can create effective neoantigens and generate robust anti-tumor immune responses. So if we take a closer look at the MHC, there's actually two versions, MHC1, which is expressed by all nucleated cells, including tumor cells, and MHC2, which is more specific to antigen presenting cells. They're fairly similar, except that MHC1 is canonically thought to bind peptides from proteins expressed inside the cell, whereas MHC2 is canonically thought to express peptides from cells that have been endocytosed from the environment. And they're both encoded on this variable region of chromosome 6 by a number of genes, and there are thousands of alleles in the population, creating a huge amount of diversity. So everyone's going to have 6 copies of MHC1, and it turns out to be 12 copies of MHC2. Sequence determines structure, determines function, so this polymorphic variation has consequences for the binding cleft for peptides and can change the specificity of binding. So now we have this model where each individual is going to inherit a set of MHC alleles. This is going to define a space of peptides that their MHC can effectively present on the cell surface, and we can intersect this with the mutations that we see causing tumors. Presumably, if the mutations are effectively presented by the MHC, they should be visible to the individual's immune system and they should be eliminated. Thus, there should be a lower probability of observing those in a tumor in that individual. Whereas mutations that are not effectively presented would not be eliminated and should easily persist into a tumor that we later detect and sequence, and thus we should see a higher probability of observing those mutations. So I'm essentially going to breeze through the first paper on this and then go to the second one, which just came out this last month. The first paper focused on MHC1, and indeed, we saw, here's the sparse matrix of observed driver mutations. This is the presentation score matrix, where each square corresponds to an individual patient's ability to present a particular mutation based on their genotype. And we see that there is indeed a relationship between the probability of observing a mutation and the ability of the individual's immune system to present that mutation. When we did further analysis, what we found is that presentation is a determinant of mutation probability. And when we look across the population, if multiple people cannot present a mutation effectively, then you're more likely to see that frequently across tumors. So the highly recurrent mutations that we see in cancer are not only functional, but they're also very difficult for the immune system to detect. So the first manuscript focused on MHC1, which is expressed by tumor cells, which means tumor cells can reduce its expression, delete it, do all kinds of things. And this would presumably be a barrier to using it as a target for immunotherapy. Well, what about MHC2? So once again, MHC2 is expressed by antigen-presenting cells, and it's a similar process. Peptides are endocytosed, degraded through the proteasome, brought into the ER, bound to the MHC, displayed on the cell surface. It's a slightly different moiety of T cell. Before we were talking about CD8 T cells, now we're talking about CD4 T cells. But nonetheless, these are key immune cells that are involved in anti-tumor immune responses. So to give you just a little bit more intuition of how the score works, we look at the individual's genotype, and for MHC2, you have an A and B chain forming MHC molecule, which results in this combinatorial space of 12 different potential combinations. So these are the 12 proteins that this person can make given their genes. We can use predictive tools like NetMHC-Pan to predict the binding affinity for peptides that overlap cancer mutations to this set of alleles. Our binding score is based on rank, where low rank is high affinity and high rank is low affinity. And so we use a harmonic mean to combine these scores because it emphasizes the lowest score, which would be the most strongly binding peptide. And then you basically get the aggregate effect of all these different MHC molecules and according to their ability to predict peptides overlapping a particular cancer mutation. In order to ensure that the score actually maps to cell surface presentation, we compare it to mass spectrometry data generated from cell lines. There's a lot less data available for MHC2 than there is for MHC1, but there is some. So if we compare peptides that were found in complex with the MHC on the cell surface to random peptides that we would expect at baseline not to frequently be effectively presented, we find that we can discriminate them to some extent. MHC2 is a little bit more difficult than MHC1. The binding groove on the molecules is open. They tend to be more promiscuous, so the assumption that a random peptide doesn't bind effectively to MHC2 is not as good as for MHC1, and so some of these random peptides may truly be binders, but there's no true negative set that we can use to really assess our performance. So in order to be able to understand tumor immune interactions, or the effect of MHC genotype on mutations, we were looking in the TCGA. The methods we're using for predicting MHC2 genotype include HLA-HD and HLA-X. We can get this for about 7,000 patients. We have a set of mutations that we think are confident drivers. These are recurrent mutations in well-established oncogenes and tumor suppressors, and so we can look and see how well each of these individuals presents the mutations via MHC2. I don't have the MHC1 results up here, but interestingly, MHC2 looks a little bit different. And I should say, actually, MHC1 is here, but it's a little more subtle. So here we have the fraction of patients presenting mutations by MHC1 versus the fraction of patients presenting mutations by MHC2. And you can see from the histogram at the top that you have a really broad array of patient frequencies. So there's really all sorts of different frequencies of mutation presentation. But when you look on the y-axis at MHC2, it's really kind of all or nothing, and usually it's nothing for these are recurrent driver mutations, right? And the highest density is down in this corner, which is just showing that many of these recurrent driver mutations are just not well presented by the human HLA. Though we did see sort of an interesting trend that some of the tumor suppressor mutations seem to not be presented by MHC2, but a lot of them were presented by MHC1. And there seems to be, as I said, a little less inter-individual variability here, but there is still some. We do notice that the presentation scores shift upwards as you select more and more recurrent mutations. Once again, this is just showing that the most recurrent mutations are those that are poorly presented. This plot here is just showing you that there is a relationship between the probability of observing a mutation and its presentation by MHC2. And this is showing you odds ratios linking presentation to probability of mutation in various tumor types, with the highest being in thyroid cancer and low-grade glioma, but these are associated with BRAF and IDH1 mutations, which we find are very, very difficult for the immune system to detect. We wanted to understand how MHC1 and MHC2 matched up in terms of the strength of effect on the probability of observing a mutation. So we did this with a multivariate nonlinear regression model. And because the scores for MHC1 and MHC2 are on different scales, we had to get the odds ratios by comparing the percentiles instead of sort of directly interpreting the coefficients. But we found that MHC2 actually had a stronger effect in terms of immunoediting of tumor mutations than MHC1. Maybe this is because MHC2 is a little harder for the tumor to evade. We find that their effects appear to be complementary, such that the highest probability of mutations are those that are presented by neither MHC1 nor MHC2. And when you look at the overlap in which mutations are presented by MHC1 and MHC2 in the same tumor, we see that they're correlated, but there is, it's not that strong of a correlation. So each of them is covering sort of non-overlapping mutations. And here we're just showing some of our favorite driver mutations. Again, this is the fraction of patients presenting them with MHC1 versus MHC2. And we do notice a few interesting ones where it looks like some of the KRAS mutations may be more frequently presented by MHC2 than MHC1. Finally, we felt that this would have potential implications for susceptibility to tumors. And we thought we could compare MHC1 and MHC2 in terms of whether they contributed to susceptibility. And the way that we approached that was to speculate that you could have individuals where their peptide space really has a large intersection with the space of cancer-causing mutations. And those people should be more protected from those mutations, whereas individuals that have a very little overlap may be more susceptible in general to developing tumors. So we looked at coverage in TCGA patients, and then we compared that to age of diagnosis. And so we have sort of high and low coverage categories, and the age of diagnosis is plotted here. And we saw that sort of pan-cancer, we saw a weak but significant phenotype. When we did individual tumors and corrected for multiple testing, we really only saw a strong effect with HCC. These others weren't significant, but you can see that a number of these tumor types look like there's an effect. And the caveat to this analysis is that we're not using a comprehensive list of cancer-causing mutations here. To really understand what the implications are, we would need to know the whole space of cancer-causing mutations. Okay, so in summary, I hope I've convinced you that MHC genotype is a determinant of immunoediting in tumors, and that not just the underlying molecular architecture, the fitness of the mutation matters, but also whether or not it's going to be exposed to this additional selective pressure from immune cells. MHC2 looks like it may have a stronger effect even than MHC1. MHC1 and MHC2 have complementary specificities. We found that MHC1 had an effect on susceptibility, but we could not find an effect for MHC2. Revisiting this with larger sample sizes, maybe we will, but as of so far, that seemed to be more MHC1 driven. But MHC1 is under direct control of the tumor genome, and thus it may be easier for tumors to evade immunotherapy via MHC1 modifications than if you have immunotherapies that are reliant on MHC2. And indeed, a number of studies have found that the neoantigens that generate effective immune responses are generating them via MHC2. I'd say a lot of what we've been talking about so far is understanding what are driver mutations and what are the mechanisms, and I just, I guess, challenge everyone to think about this aspect of tumor evolution as well, and how that might affect your conclusions. And by the way, this happens in mice too, except that we have very specific mouse genotypes associated with certain strains. And so one driver mutation may appear in one strain more easily than it appears in another strain. So that's something to think about. Okay, so I'll stop here and tell you that this work was done by a fantastic former graduate student, Rachel Pike, who is, as of last Friday, Dr. Pike, and in collaboration with a lot of folks, including Trey here. And I'd like to thank Trey and Nevin for putting on the symposium and inviting me to speak. And with that, I'll take any questions. Thank you very much.
questions?
I have a naive question as a non-immunology person, but how would this affect CAR T-cell therapy in a way? I mean, does it mean that they're going to have to think about whose T-cell and what the MHC locus is in the T-cells that are being used, et cetera? Does that make any sense?
Yeah, so I think that the deal with CAR T cells, though, is that you engineer them to be specific to various cell surface markers, and they become independent of MHC1 and MHC2 in that sense. Sure. I think one of the challenges with CAR T cells is if you have a cell surface marker, and I'm just going to say CD34, although I'm not sure if that's the right one. If it's a single marker CAR T cell, then the tumor cell could stop expressing that cell surface marker, and then it would immediately become resistant to CAR T. There are some people that are trying to engineer sort of more combinations of molecules, so CAR T cells that can detect multiple cell surface markers simultaneously, which would make them harder to evade.
So you said that mice, which we have all these inbred strains of mice with the same MHC, present peptides differently, but then there are humans also who are homozygous, right? And do people with lower MHC diversity have increased susceptibility to cancer or cancers?
We definitely see an earlier age at diagnosis. We haven't validated that it's a total susceptibility effect. We know that with age, the immune system becomes compromised, and we see a very strong age-associated difference in strength of immunoediting. So it may just be an early versus late question, but you could certainly imagine, and we're trying to pursue that question.
Thanks, that was great. I was wondering about pathogens, and those will also present antigens of pathogens. Is it known that some people are more susceptible because of the variants they have? MHC class variants.
I think that's a fantastic question. So we haven't obviously applied this directly to pathogens, but I think it's pretty well established that the MHC genotype is in a evolutionary battle with pathogens. So pathogens are trying to evolve away from them, and they're trying to catch back up. And that's really what drives the diversity of MHC in our genome. In fact, it's not designed for cancer, and it's actually pretty bad at cancer, but it's doing something that's helpful, right? So I think that's a fantastic question, and I think we could apply this framework to investigating questions related to pathogens as well. But yeah.
Thank you very much.
Great, thank you.